Welcome to Lesson 8 of Practical Biostatistics. Thank you for viewing. To the viewers who have subscribed, thanks a million. And for those who haven't subscribed, please do so. We have looked at comparative statistics for one group, two groups, and more than two groups. For today's lesson, we will be looking at association statistics, especially correlation analysis. To determine whether there is any association among paired variables, that is, variables measured at the same station, measured upon the same sample, or at the same time. For example, when measuring bacterial abundance and chlorophyll A concentration at each sampling. Therefore, at the end of the research study, we should have a paired data of bacterial abundance and chlorophyll A concentration. In parametric statistics, we use the Pearson product moment correlation. From the correlation analysis, we will obtain either a correlation coefficient, as indicated by a small letter r, or correlation index, as indicated by a capital R squared. Correlation index is also known as coefficient of determination. I prefer to use correlation index in my writing and analysis, as it describes the percentage of variation in the y-axis that can be explained by changes in the x-axis. For example, when you have a correlation index of 0.4876, when you plot chlorophyll A against bacteria, then you can state that the variable chlorophyll A explained 48.76% of the variation observed in bacteria. We log transform the bacterial abundance in order to use parametric statistics. Here we see that correlation coefficient is able to show direction, whether positive or negative. For example, if positive, that means log bacteria increases as chlorophyll A concentration increases. In contrast, if correlation coefficient is negative, that means log bacteria decreases as chlorophyll A concentration increase. An important point to remember when using the results from correlation analysis is a significant correlation does not mean that there is causal relationship or a cause and effect relationship. Usually, a significant correlation necessitates further investigation, maybe through laboratory experiments, where you can manipulate one variable and then you measure the response of the other. So this will prove that the correlation that you observe is a true cause and effect relationship. However, you may still use significant correlation in your writing if the relationship is a well-established relationship. In other words, well published and accepted by the scientific community. For example, the coupling between bacteria and chlorophyll A, that is a relationship that is generally accepted by the scientific community. But for other pairings, please be careful how you use them, as sometimes spurious correlation occurs. For example, in field studies, when chlorophyll A and bacteria are both influenced by an external factor, for example, a nutrient source. Therefore, the correlation that you observed between chlorophyll A and bacteria is actually just a co-variation of both chlorophyll A and bacteria against the nutrient. However, if P is above 0.05 in carrying out correlation analysis, then there should not be any causal relationship between the pair of variables analyzed. So, significant correlation does not mean causal relationship, but correlation is a necessary ingredient for any causal relationship. This is an important point to remember. In correlation analysis, when the number of samples used is high, maybe more than 30, then it becomes easier for the correlation to be significant. Therefore, you may use the table here as a guide to determine the strength of your correlation, especially when the number of samples is high. You'll have R squared when the relationship is considered weak, moderate, and strong. the source of this table is from Pearson. Once you have determined the R and the R squared, you can use this equation to determine the student's t value. And then from the student's t distribution, you will be able to find your probability. Finally, the non-parametric statistics equivalent to Pearson correlation is the Spearman's ranked correlation. Now let us try out correlation analysis in Excel and pass. So here we have paired data sets collected from a coastal water location, Port Dickson, over a period of a year. First thing we have to do is log transform the bacterial abundance in order to use the parametric statistics. Then you can drag the equation to cover the area of interest. Then for chlorophyll A, we will just copy and paste it here. The order is as below. Excel will treat the first column as X and the second column as Y. So let's carry out correlation analysis in Excel. Under data analysis, look for correlation here, and then the input range, you highlight both the columns and then tick the labels in the first row, and for the output range we can put it here. Okay, so in Excel, when you carry out correlation analysis in the analysis toolpack, what you obtain is the correlation coefficient, but it doesn't tell you whether this is significant or not. So the manual approach to this is we can start by getting the count data to determine what is your n, number of paired samples. Then to determine the correlation coefficient, we can type the following formula: correlation, and then select the array 1, and then array 2. Okay, so this is the correlation coefficient. Of course, if you want to obtain the correlation index, you can just square this to obtain your R squared. And now let us use the equation in the slides to calculate the student's t value. So need to get the R times the square root of n minus 2 divided by the square root of 1 minus R squared. Okay, so this is your student's t value. So finally, we need to determine whether this value is significant. We will be using the t distribution, two-tailed, with the t value here and the degree of freedom, which is 10. 12 minus 2. So the degree of freedom in correlation analysis is n minus 2. With that, we can get a probability of less than 0.05. That means the relationship between these two variables are significant. So let's use pass to try it out. What we can do is we'll just copy these two columns and then use pass, select column attributes so that we can paste the label. Once you've done that, Control A to select all, go to univariate. There's a correlation menu here. Tick, and you can see immediately that your results are available. Okay, so this will be your correlation coefficient, and this will be the p-value. The good thing about pass is you can also carry out the Spearman's rank correlation here and obtain the Spearman's RS here. Thank you for watching, and please remember to subscribe.